Yes, so my presentation is about Cibacter and lignocellulosic biomass processing possibilities in Latvia. It was also called like a waste-free rational use of fruit trees and shrubs lignocellulosic biomass. Sorry. Yeah, now I see. Yes, so here we see the Cibacter. Okay, maybe it's not timber, but it's still the wood, the tree, which we can use. So the question is why we should use the wood of fruit trees. So we consider that it's small gardens or something, but no, it's like the wood from fruit trees in the world is the amount of area of forest in the EU estimated as 135 million hectares, and the area of fruit trees in Europe occupies total of 11 million hectares. So it's huge. In the same way that the forest needs a sanitary cuttings, fruit trees also need forest and plant pruning, which results in high and large amount of food waste. Garden trees without shaping and care give low yields. Cibacter as a nitrogen-fixing pest-resistant deciduous shrub tree, which grows in Europe and in Asia and also South America. And according to the report in 2023, it grows in 52 countries with total area of 3 million hectares. In Latvia, it's very important because it's the second most common fruit tree species after apple, and one of crops whose planting area is constantly increasing. There are about 400 hectares of Cibacter, and about 250 of them are in age of harvesting. Global Cibacter berries export market is valuated at $2 billion US. So the most popular varieties in Latvia are Maria Brugale, Tatiana, Botanicheskaya, Любительская, because of they have larger and juicier fruits. Why we should use the wood of Cibacter? It's a very interesting tree. First of all, because of it survives the extreme temperatures from here is not written, from plus 40 to minus 40, and which forces the plant to develop adaptogenic qualities and evolve with this diversified chemical portfolio. Cibacter belongs to prolific ancient species, and almost all vitamins found in nature are found in Cibacter. A large volume of cut Cibacter branches is formed every year because of harvesting of the berries in summer and autumn or seasonal pruning. Usually it's early spring. Cibacter tree should be almost completely cut back every 4 years. And the industry of uses fruits, but there are almost no application found for the other part. You can see on the pictures, here is the orchard, and here are the cut branches and cut branches which are cut together with the berries. Due to the strong and vegetatively developed root system of Cibacter, it's also used in land reclamation because of its nitrogen fixing. A little bit of history, we can see that since already old times, people should cut the Cibacter, and otherwise it's not good to harvest it. It's too high and it's not useful to be so people can pick up the berries. So relevance of the study for the rural economy, it's because of the great competition of Cibacter and berries market, it makes us think to use about usage of all plant parts. And Cibacter and lignocellulosic biomass contains a variety of available secondary metabolites, and they include proanthocyanidins and serotonin. And it's necessary to develop the no residue scheme for the Cibacter production. And so this new approach, complex and rational use of the whole fruit tree, can help to promote regional development, can help with the work during non-seasonal periods, can create new jobs, and selling side products would give additional profit. So the aim of the study was to propose wasteless innovative scheme providing usage of all unique pool of biologically active compounds extracted from non-fruit trees and from fruit trees, and an example of Cibacter biomass in form of extracts, individual compounds. In our case, we studied proanthocyanidins and serotonin for creation of multifunctional, safe antimicrobial and anti-inflammation formulations for human and animal care, and solid granulated complexes for remnant of feed and plant growth. We also, at the moment, are studying in our new project the Aronia melanocarpa biomass, and we will show a couple of results from Aronia biomass to prove that proposed scheme for Cibacter could be useful also for actually maybe any fruit trees. We can't tell now any, but at least our Aronia results confirm that it's possible to use it for Aronia and other fruit trees. So the following task has to be set: to assess the potential of biomass and to extract serotonin and proanthocyanidins, to find the best conditions for this extraction. We also checked innovative extraction with Freon for using as antimicrobial and anti-inflammation means. It's necessary to determine cytotoxicity, effect on hemolysis, and to detect also antimicrobial activity by itself. We also checked the activity, the influence on activity of digestive enzymes, and we also checked for closing the cycle of biorefinery. We also checked the post-extraction residues as raw material for production of granular fodder and plant growth promoter. And such a scheme we should propose to the growers. So why Cibacter again? It's because of we know that Cibacter berries contains more than 190 different biologically active substances, almost all known vitamins. So if berries are so available, what about the other parts of the tree? So it was very interesting to check. So what about leaves? Leaves are a little bit more studied than, so the twigs are almost the branches, so almost no study, but leaves have some ancient formulations for teas, and they also have some studies which show that complex of chemical composition and pharmacological properties opens up opportunities to study them as the raw material for production of wound healing, anti-ulcer, and vitamin preparations. So the potential of Cibacter for extraction of biologically active substances, we see that one of the very good compounds are polyphenols, which accumulate during the process of plant growth and development, providing plants with important physiological and morphological properties, protecting the plant from biotic and abiotic stresses, and providing protection against infectious disease agents. So previous our studies showed that proanthocyanidins are the dominant compounds in oligomeric polyphenols obtained from lignocellulosic biomass, wood and bark of deciduous and coniferous trees. Why proanthocyanidins? Proanthocyanidins in plants represent the first biochemical defense to external injuries and infections. Proanthocyanidins have been at the focus of increased interest in the last years as a result of high biological activities, antioxidant, antibacterial, anti-inflammatory, anti-cancer, which have prompted their use in therapeutic needs. So, but proanthocyanidins from Cibacter woody biomass were not studied. Proanthocyanidins, as commercial source of proanthocyanidins, they are also called condensed tannins, include quebracho, wood and bark acacia, and many other trees which are studied already. And they are widely used for tanning raw hides in the leather industry, as well as adsorbents for water purification. They also were studied also by our colleagues in the production of glues in wood industry and in production of anti-corrosion agents. The use of Cibacter and proanthocyanidins or extracts containing them for industrial purposes, for example, for wood adhesives, is probably not economically viable due to the large volumes required. Therefore, we choose as application directions with a highest added value, a biological activity of proanthocyanidins and also serotonin. I will tell about it later. So we choose this direction as with the most added value. Chemical structure of proanthocyanidins you can see, so the different forms of them, A-type, B-type. Serotonin, why serotonin? Serotonin content have been shown to vary at different plant parts, and it's not so often to find it. So it's quite, the studies of serotonin are quite new, and serotonin contains also in humans, and mostly in brains, but also in the gut. So the physiological functions are not so clear in plants, but it's already found that functions such as detoxification, iron permeability, antioxidant, antipredator defense, and so on also are controlled by serotonin. It also protects plants against fungi. So it would be very interesting to study the effects on humans. And the amount of serotonin was found in the berries around 30 micrograms per gram. And as to berries, so 50 grams of berries are needed to provide serotonin levels. So the formula you can see, structure formula, and 95 of serotonin is produced in peripheral organs, and serotonin in digestive system can act independently of serotonin in the brain. Now it's so-called gut-brain axis, which connects what is in the gut. The latest studies are showing that what is happening in the human gut could happen in your brain. It's not so directly, but it influences it very much. And why? So we choose, as I told, we choose our direction as one of them to test on antimicrobial activity. Why is it like that? Because of number of deaths related to antimicrobial resistance from pathogenic bacteria are growing constantly, and the resistance is absolutely awful, and doctors are often telling that this will be a huge problem if in the hospitals the most strongest antibiotics will not work. So we need first of all to not to use antibiotics so much. We try to use them only when it's necessary, or to help with natural compounds, or to replace it if possible. Why anti-inflammatory activity? Anti-inflammation is a protective function, but excess inflammation can induce host tissue damage, as for example, the latest now is known about COVID. And it also causes even cancer. Currently, the anti-inflammatory drugs are mainly steroid and non-steroid, but they have frequent clinical side effects. So the alternative is necessary. Proanthocyanidins in plant represent the first biochemical defense to external injuries. So the studies have demonstrated the anti-inflammatory activity of proanthocyanidins. So here we show the scheme of what we wanted to study. So we wanted to evaluate, first of all, the different species. So then we wanted to evaluate the differences between summer autumn biomass, so the branches which were cut together with during the harvesting together with berries, because for industrial collecting of berries is the only way possible. And the another part is winter spring. Then we evaluated different types of extraction and optimization of it. And then we evaluated antimicrobial, anti-inflammatory, antioxidative properties, influence of digestive enzymes, and the remainders of the extraction we tested for animal digestibility and for plant growth and development, which shows us possibilities for medicinal and cosmetic formulations, food additives, animal feed, and plant growth stimulators. The work was done in cooperation with Latvian University Faculty of Biology, Riga Stanich University, Faculty of Veterinary Medicine in Bulgaria, Bruvel, and the Composite in Ltd. So the results show that the general characterization was done by analytical pyrolysis, and the results show that branches in leaves contain polyphenols and containing compounds that indicates the presence of secondary molecules. If you remember the formula, that they could be serotonin and proanthocyanidins. So the potential of in order to find the extraction conditions, we tested different ways, different temperature, concentration of ethanol, and found the optimal. And for the extracts obtained using the same solvent, the proanthocyanin values for different species were close to each other, cultivars, were close to each other for all Cibacterium cultivars growing on the same plantation, which indicates the similarity of ability to synthesize secondary metabolites. So for us, we choose for the further studies the three most prospective cultivars. So it was Maria Bruvana, Ботаническая любительская, and Tatiana. And with the highest contents of proanthocyanidins and serotonin were for Cibacterium branches extracts obtained with 50% ethanol solution. The most promising was Maria Bruvana. And this shows that for more efficient extractions, To increase the yield, it is necessary to separate the leaves, and separated leaves were tested for as preservatives and for lipids-based systems and as feed additives. So, empirical chemical characterization showed that that content of protein highest in the 3-year and 4-year branches, while the serotonin content is in 1- and 2-year branches. Seasonal variations show that autumn is the most suitable for the production of for the extraction of serotonin and proanthocyanidins. And the highest content was determined in the bark, but economically peeling of the bark, we tried it, so it's not so easy, and that's why we offer unpeeled 1- and 2-year old branches with the serotonin content in the extract is the highest. So we made the characterization of proanthocyanidins and found that catechin dimers and trimers are there. And innovative freon extraction was tested, but it's not so good for rural development. So we just tell it that we tested it, and it didn't show the best results. This is for lipid-based stability systems. So antioxidant activity was tested, and it showed that a relationship was observed between the contents of proanthocyanidins and extract and their antioxidant activity. The highest proanthocyanin content, the radical deactivation increases. So among the own extracts, 50% ethanol extracts again show the highest oxidant activity compared to aqueous extract. And as I told, we also tried a little bit for aronia already, and really it also showed the same, even better results than compared to Cibactone, and better results that gray algal, for example, from our previous studies. So we tested bioxypress method. It's increased with increased temperature and increased temperature. We simulate the so it goes faster. Yes, all the oxidation goes faster in this equipment. So we tested the ability to delay oxidation on lipid-containing products, and we found that 50% ethanol extract, 50% ethanol extract is most effective, and the leaves showed same activity as the water extract. So the extracts were better in the system with a lower lipid concentration. And then we tested the antimicrobial activity of the extracts and serotonin-rich fraction, which showed that purified proanthocyanidin and 50% extract with higher proanthocyanin content than in water extract had the best antimicrobial activity against all the bacteria except for Bacillus cereus. Serotonin-rich extract was less active, and but the antibacterial of the serotonin-rich extract, again, Cutibacterium acnes, was even a little bit higher than that of pure serotonin, and that's why we considered at the moment we considered the purification of serotonin-rich fraction, but we decided that it's not necessary to purify it more than 28% as we did, because of it used high activity than pure serotonin. So we again tested the aronia already, and we can see from the results that the results of antimicrobial activity and antifungus activity, no, I don't see here, sorry, antifungus activity, but antimicrobial and antimicrobial activity, no, we have Candida albicans. So they are also very good for aronia. So the potential of the tested proanthocyanidin for further development of antimicrobial, antifungal applications is very good. What is important? So if you use something for antimicrobial, antifungal activity, we need to know if it will not kill our cells. So we tested the cytotoxicity on human cells, and in most cases, the concentration similar to inhibitional concentration and killing concentrations. In all antimicrobial activity tests, there were no toxic effect, so we can use the extracts and proanthocyanidins. Then we use checked anti-inflammatory activity. It's for IL-8 secretion in unstimulated and stimulated blood mononuclear cells, and we can see that plant proanthocyanidins can reduce inflammation And we stimulated polyethylenic and polyethylenic acid, we can see inflammation which is caused by viruses. So we can see that it also can reduce inflammation connected with viruses, caused by viruses. So we also tested on red blood cell hemolysis. We tested the red blood cell destruction, and none of the extracts induced hemolysis after 1 or 8 hours incubation. That indicates the high biocompatibility and safety of the extracts. So as I told before, we wanted to close the cycle, so we tested both the biomass and residuals after extraction for possibilities to be used as an animal feed, and we checked the in vitro gas production and digestibility of Cibacterium biomass in experiments with small ruminants digestive system. So according to in vitro test data, the extract showed the greatest digestibility after the separation of condensed tannins, or proanthocyanidins. The leaves have a much higher digestibility than stem. These results confirm that it's better to separate proanthocyanidins, and also that confirms that it could be very good to add leaves in all formulations. Here we can see the results and the photos of granulated biomass. So what could be one of possible schemes for rural development? We can take the twigs, we can make extraction. We have extract contained condensed tannins, so proanthocyanidins, which we can use as antibacterial, anti-inflammatory means, and then we can use twigs biomass as a residue, for example, for fuel granules, but we also can use more valuable applications, stem biomass extraction residue for granulated animal feed. Yes, and with various mixtures. So we made also the characterization and found that there are also a lot of vitamins, same as in the berries, and micronutrients. And we checked for the heavy metals; they didn't exceed the amount which is demanded by European Union. And we also tested the granules for swellings, because of these granules, which are for feed, they need to swell in order to be better fed to the animals. And the animals say them very well. Which tested also extracts and proanthocyanin on pancreatic lipase activity. And the results were interesting because of the proanthocyanin showed inhibition of in one dose, but also they showed activation in another dose. So they can be used in different applications. For example, inhibition is necessary for people who want to be not to eat so much, but the increase of activity need for people with malnutrition. So there is another scheme of how we can use the residues, but I already think I told about that. So what are our conclusions? That Cibacterium lignocellulosic biomass, which is formed as byproduct as a result of berry harvesting and as a waste result of shrub trimming, and new is a valuable raw material for production of proanthocyanidins and serotonin. And as regards to proanthocyanidins, we can tell it also about aronia already. Autumn is the best seasonal period for the extraction, and the antimicrobial activity was proven for variety of bacteria. So there is a possibility of replacing or used together with antibiotics. And the range of in the range of antimicrobial activity of extracts and proanthocyanidin, was no cytotoxicity. And hemolysis tests also showed good results, showing the high compatibility or safety of the extracts. The proanthocyanidins have high anti-inflammatory activity, and proanthocyanin extracts have also high antioxidant activity. The target compounds have also the high antimicrobial activity against pathogenic bacteria that contaminate skincare products. So we can use these our extracts from proanthocyanidins as a multifunctional product, multifunctional active additive that will be used, for example, in cream, not just like antimicrobial ingredient, but it shows also the anti-inflammation activity. So it's also very good that it's with different functions we can replace maybe different 3 ingredients with 1. And it has been determined that due to anti-inflammatory properties and non-toxicity, target compounds are a promising source for production of multifunctional products for use in medical, topical formulations, and cosmetic. And they can be used also in food additives. Chemical characterization and in vitro analysis in the digestive system showed possibility of use in animal feed, and also the different inhibitory and activating effects of target compound, activity of human digestive enzymes with acetylcysteine amylase and lipase have been shown, which will also allow to use the study compounds for various digestive problems. A waste-free scheme was elaborated. Here is the waste-free scheme, so we can see that we take the butter and we use the berries already uses elaborated before us, so we didn't study it. Same with biomass, and we studied and elaborated the part for ligand cellulosic biomass. And you can see on this scheme that it's also circular, so we can, for example, plant growth stimulators, we can return back to the bacteria. And we elaborated also the scheme for growers, which will be very good for rural development, because we can see that growers can, from this scheme, they can use it to see what they can get from Cibacterium, or Aronia, or possibly other fruit trees. So all the tree is a very good system. It gives us everything we need. We acknowledge the two projects in France of which this work was elaborated. Thank you for your kind attention, and our coordinates for questions.